Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Moving the Needle in Vaccine Trials, Optimizing Digital Endpoint and Operational Strategies to Enable Study Success. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and a broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 30,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 70% of all FDA drug approvals since 2012. Now, it is now my pleasure to welcome our speakers to today's event. And first, I'd like to welcome my co-moderator for today's event, and he is Dr. Todd Rudeau. He is the Executive Vice President and Chief Medical Officer at Clario. Hello there, Todd. He's providing medical and scientific leadership across the organization. He has nearly 20 years of clinical cardiology and pharmaceutical research experience with a career predominantly on drug safety. His team provides expert consulting to clients on scientific and regulatory strategy and ensures Clario's product portfolio is scientifically robust, generating high-quality clinical trial endpoint data. Next, I'd like to welcome and introduce you to Saima Kakwani. She is the scientific advisor at Clario, hello there, and the internal subject matter expert for vaccines. She specializes in eCOA design with a focus on reducing patient burden and improving data quality through best practices in diary design, including the use of paper and free text. With over 6 years of experience supporting phase 2 and 3 trials, across multiple therapeutic areas, she has led scientific contributions in oncology, respiratory, pain, diabetes, cardiac disorders, and vaccines. Now, finally, it is my pleasure to welcome Dr. Fabian Chan. He is the Deputy Chief Medical Officer, Cardiology, and Vice President, at Clario. And there he is. Hello there. In addition to providing leadership for Clario's Cardiac Safety Department, Dr. Chen offers expert consultation to clients on scientific and regulatory strategy. Dr. Chen has nearly 20 years of clinical cardiology and pharmaceutical research experience, and he also has over 10 years of experience in both early and late stage clinical development. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Todd. So Todd, when you're ready, you may begin.
Great. Well, thank you, Sonia, for the very kind introductions. Thank you to all of you, our quite sizable audience that has joined us today as well. I'm particularly excited about this one. There's quite a bit we've packed into the session today, focusing on meaningful insights to optimize strategies within vaccine development programs. So We'll take you through, you can go ahead to the agenda, please. We'll take you through this agenda today, starting off with just some general comments from me on the challenges that are especially unique within this therapy area. Then Simon will spend the majority of our time speaking to optimal diary design, safety monitoring and vaccine programs, considerations for what ePro technologies may fit best, depending on the study objectives and populations, considerations for patient burden and compliance. She'll also spend some time on the regulatory perspective, so strategies to meet program objectives while also hitting the mark from a regulatory guideline or requirement standpoint. And then Dr. Chen's going to take us through some of the unique elements for cardiac safety monitoring in vaccine development. This does vary by program and by vaccine type, and he'll cover a bit about COVID vaccine-related myocarditis as well in the end. I'll wrap us up for the Q&A session. Please do send in your questions over the course of the webinar. We'll definitely leave some time at the end to get to them, so look forward to that part of this as well, certainly. If you can go to the next slide. To get an understanding of this audience's perspective, we asked you all during registration and on LinkedIn as well, what do you see as the biggest challenge to a successful vaccine trial today? So digital endpoint strategy, global operations and site readiness, patient burden and experience, and then safety monitoring. So you see the results here on the screen. Generally, this audience was fairly evenly split, seeing all of these elements as potential meaningful challenges. By a slight margin, the greatest concern, though, was managing patient burden effectively, followed closely by operational concerns. I'll say that this even split doesn't surprise me. It makes sense. These in totality do represent the key considerations. So we'll be sure to touch on these throughout the next hour. So with that overview, you can go ahead to the next slide. I'm going to take a few minutes here just to set the stage, highlighting some of the unique elements of vaccine development as a therapy area. I'm starting with this as I think it's important context to understand the why and the how, so the solutions and strategies that have evolved over time. In other words, what's difficult in this space and how are we addressing it? So on the right, that figure highlights not all vaccines are the same. And while the objectives of preventing an infection or reducing the severity of an infection are aligned, how we achieve this mechanistically varies quite a bit. So inactivated virus versus live but attenuated virus vaccines, the unique mRNA vaccines, which rather than injecting an antigen into the body to mount a response, these enabled the body to actually generate both the antigen and the immune response. Synthetic peptides, for example, you're familiar with them, and some of the others listed here as well. These different classes can have different safety considerations, and so the solutions that we deploy in a study can vary, accordingly. The phases of development are the same as those you're familiar with in other TAs, with some nuanced differences, though. The discovery or preclinical phase is focusing on, which antigen or identifying the best antigen to go after, managing formulation, beginning to understand toxicity, leveraging in vivo and in vitro models. Phase 1 healthy volunteer studies are mostly about assessing safety as with other therapy areas, but they may include early elements of assessing immunogenicity. That's what is the body's response, immune response. Phase 2 studies, a bit bigger than phase 2s in other therapy areas, so hundreds or even low thousands of participants here typically. This is about dose optimization, understanding immunogenicity, extended safety monitoring as well. And then phase 3 is, often tens of thousands of patients in the target populations, focusing on demonstrating efficacy in that case. These phase 3s, of course, are a bit longer and more expensive studies than other areas. Post-marketing pharmacovigilance is also an important element in vaccine therapy, executed in a number of different ways, whether it's registries or formal phase 4 studies. But that's a big part of vaccine development as well in terms of monitoring safety in real world use. So what's unique? Well, let's start with the fact that large numbers of healthy people will be taking these as a preventative measure. These are not sick people that are being treated for a disease. They're healthy, but at risk for a disease. And in that setting, the risk tolerance for the drug is quite different. The safety profile of a preventative therapy must be better than the safety profile of many therapeutics that are intended to treat a sick patient, with a particular disease. Immunogenicity, so as I said, that's how the body reacts to exposure to an antigen. We can measure that quantitatively. So for an antibody-mediated mechanism of immunity, we can measure quantitatively the amount of antibody response. To an extent, we can measure T-cell response for conditions that have T-cell mediated immunity. But what we measure, and this is the challenge, what we measure in the lab, that immunogenicity correlates differently in different diseases to actual efficacy. So that antibody response doesn't mean necessarily that it's working, essentially. So the unique challenge here is navigating this and trying to understand what the correlation is for a particular disease. Trial size, large by necessity. Often a great deal of participants are needed to be exposed in order to understand its efficacy, particularly if the incidence of the disease is low. You also need a large number of subjects to detect rare safety events. Compliance, so Simon is definitely going to spend some time on this one. The issue here is that if you receive a dose of a vaccine and you feel fine, there's a risk you simply stop responding to any trial-related activities, right? And that leads to missing data, which is very problematic because missing data cannot be just assumed as negative data. And so appropriate and sort of low burden technologies deployed for the participants in a trial are key here to maintain that compliance and retention over the full protocol. Some other highlights that are significantly different: manufacturing biologic products at scale has a host of unique challenges. Regulatory expectations, again, we'll touch on that, but there are some specific requirements here, often including special high-risk populations. A particular challenge, perhaps even more so in today's current environment, is public perception and vaccine hesitancy. So this leads, of course, to recruitment and feasibility challenges when running a study. And then lastly, expedited development pathways may be necessary. And we saw this, of course, during the recent pandemic, but these expedited pathways still come with the same responsibility for robust safety monitoring. And so, the additional challenges of doing that in a rush, so to speak. So on the next slide here, I will hand over to Saima, who's actually joining us from Amsterdam, as it turns out. She's presenting at the World Vaccine Congress this week. So as you heard, Saima is a scientist in our ECOA consulting team, science and consulting team. She's going to take you through some of the challenges I just highlighted and speak to how the right PRO strategy and technologies can help address them.
Great. Thanks so much, Todd.
Yep.
So as Todd outlined in his introduction, where vaccine development brings its own set of unique challenges, so we need to think a bit differently when we are implementing any sorts of vaccine trial in no matter what geography or what population you're looking at. And what I want to specifically talk about is safety monitoring and participant compliance. And to start off with that, I just want to give a brief context of what clinical outcome assessments are, because they're extremely critical when you're looking at vaccine clinical trials. So we just don't want to look at antibody titers or lab values when you're looking at vaccine trials. We also want to understand how participants are actually experiencing the vaccine itself. And your clinical outcome assessments can actually help build that bridge between the science and also the participant's voice. So it can help capture things that you can't measure in a lab. So for example, did the participant face any fatigue? Did they have any pain at the injection site? How their daily activities were affected post-vaccination? So this can help you give you that participant voice, and then it also helps strengthen your safety monitoring. Within clinical outcome assessment, we want to look at patient-reported outcomes. So these are tools that participants use to report their symptoms, and this can help you get data in real time. So as participants are reporting their symptoms, any reactions to the vaccine, so rather than relying on memory, so when you see them at the next site visit, we can actually embed this tool within your trial so you get real-time data. And for vaccine trials specifically, the type of patient-reported outcomes that we use are reactogenicity diaries, and I will go into that detail in just a bit. And at the same time, you have your regulatory bodies such as your FDA and your EMA that are constantly emphasizing on patient-reported data, which should also be standardized across your multiple phases and across your multiple studies in vaccine trials. And your clinical outcome assessments can actually help ensure that the data that you're collecting across these studies, across phases, it's consistent, it's structured, and it's also submission ready at the end of the trial. And with this consistency, it can help improve your data analysis at the end and can actually even help accelerate approvals. So what we want to look at, that COAs are no longer something nice to have, and they're just not like optional extras anymore. We want to ensure that they're embedded in a way that they make your vaccine trials rigorous, they give you reliable data, and your trial design is actually patient-centered. So now let's look at how we can do that in practice. Next slide, please. So building on that, I want to now zone in further on the reactogenicity diaries. So your electronic or e-diaries are one of the most powerful tools that you can use to capture your clinical outcome assessment data. And they're not just something that you used to use paper and now you're doing an electronic implementation of it. However, they actually fully transform the quality of data that you get for your trial. So how does it do that? E-diaries first give you high quality data as they're time stamped, and they're also date stamped. So this ensures that participants are actually recording their symptoms and reporting their symptoms in the correct time visit windows. This is absolutely crucial when you're looking at safety monitoring. So you want to track solicited reactions in the days immediately after vaccination. Also, we can use preset response parameters, any on-screen prompts to help guide participants and reduce the risk of errors. And if you have a participant who has missed a single entry, you can have automated alert flags as well, so the site can quickly follow up. The second advantage of having an electronic diary is that they enable real-time data. Investigators and sponsors can review the diaries as they are submitted, so they see what the symptoms are being reported by the participant in real time. So again, you're not waiting until the participant comes to you at the site visit. You can monitor these in real time, so your safety signals can be identified much sooner and then can be acted on faster as well. And because your e-diary data can be linked with your other data sets, such as you can link these with your lab results, for example, or any wearable devices, your analysis can be much richer and quicker as well. And lastly, the data that you get at the end of your trial through these e-diaries is submission ready. So unlike paper, with electronic, there is no risk of transcription errors, and the data then also aligns with your global regulatory guidance. And the data that you're gathering, it's flexible enough to adapt if your requirements evolve in the future as well. So it's not just about just convenience, but also looking at how we can make participants more compliant to participate in the trial, but also their diaries are embedded into our scientific validity. And how do we ensure that we keep science at the crux of developing vaccine diaries? Next slide, please. So we don't want to do an electronic diary and just checking a box that this is done, this is implemented. We want to ensure that we're asking the right questions from the participants at the right time. And this is where the science of your e-diary design comes into play. So what kind of questions you're asking, so your wordings are very important. The scales that you're using, which are your response parameters, and all the different response options that you include in your questions. These all influence how your participant responds to the electronic diary, and then also the reliability of the data that you will be getting. So for example, simply just asking the participant post-vaccination, did you have a headache? This will give you very limited insight into the participant experience. What the regulators also want to see is just, it's not just the presence of headache. So was there just a minor annoyance for the participant? We want to look at how it interfered with their daily life. What was the impact of this headache, and what was the severity as well? Because if it's a severe disruption, that is critical to know for your vaccine trial. Also, we want to ensure that the type of diary that we're designing aligns with the guidance from the FDA and the EMA using the toxicity grading scales. I'll go into detail in a bit as well. Where we look at severity and we look at the functional impact, because we want to ensure we get this data to determine how your event, i.e. let's say if it's a headache, how that should be classified. So it's not just a technical task, it's not a checkbox exercise. It's actually a scientific activity to design a very meaningful e-diary that is fit for purpose. So if you get this right, you can then capture meaningful data, you can get actionable data that can reflect the true participant experience. And if you get that wrong, from the get-go, we actually risk missing or misclassifying any safety signals, or you could then undermine the entire trial data altogether. So what are the different considerations that we should keep in mind? Next slide, please. So when it comes to designing e-diary specifically for vaccine trials, there are 3 areas that you should focus on. The first one is design essentials, the scientific rigor, and usability with engagement. So for design essentials, and we hinted on this in the previous slide as well, we want to ensure that our diaries are focused on the local and systemic symptoms that are expected by the regulators, that they already want to see because the guidance has already been provided. So you're measuring symptoms such as injection site pain, you're looking at fever, you're looking at fatigue. So for an e-diary, because the guidance is already there, we want to ensure that we use that and create a simple diary. So simplicity here is very critical. Even when you're using scale, for example, if you're using anything for headaches, you can use none, mild, moderate, and severe. Again, we want to make it as simple as possible for the participant. We can also add definitions. So for example, if you're thinking about asking, if you're including redness or swelling, you can also define that for the participant in the diary. We don't want the participant to guess what this could potentially mean, or they try and infer the meaning of it. We want to be as clear as possible and as direct as possible in our questions. And then we align that diary completion windows with the known reactogenicity timelines. So for vaccine trials, that looks like 7 to 14 days post-vaccination. After the 7 days, you can ask the participant if they have any symptoms to report, and you keep asking that up until day 14, or when the symptoms are most likely to disappear by. Then your scientific rigor. So every item that you include in your diary is embedded in your regulatory guidance. So these guidance have been provided by the FDA and the EMA for how these assessments should look like for your vaccine trials. You can also pilot your diaries in a certain population, the population that you're looking at, to ensure that the participants fully intend how this is supposed to be used and how they are going to answer these questions. And then again, as you move forward towards multiple studies, different phases of the trials, you can, it's recommended that you apply standardized terminology across the studies. So your data that you're gathering from across all these studies from your different phases, it's comparable, and that will just give it more scientific rigor at the end. And lastly is the usability and engagement, because we want to ensure that if you're asking the participant to complete a diary for 7 days, we want to ensure that they're fully engaged for the entire duration, and they're completing the diary on time every day as it's expected. So what we want to do is we want to ensure that we provide clear instructions and examples. So again, what counts as redness when you're looking at site, injection site, what counts as swelling, for example, how to differentiate between the two. And then a very important element of ensuring usability and engagement is your built-in reminders and alerts and notifications. So don't rely on participants' memory. Oh, I think I should complete my diary now. Have your reminders and notifications built into your tool that will allow participants to remain engaged, and also they're not relying simply on their memory for completion of the diaries. And then on the next slide, I want to show you a real world example of why it's important to ask the right questions from the participant. So we're asking the participant, how much have your headaches interfered with your normal activities? And the first option says no headache. And then we ask the impact of the daily activities. Then in the next question, we are asking about their muscle aches. Again, how much it has interfered with their usual activities. So at first glance, it looks okay, detailed. You're asking about headache, you're asking about muscle aches, but there's some real issues here that can compromise your data quality. First, these questions are asking about different constructs, but they have been combined into one question. So the headache question also looks at the presence of the headache, but then also the impact on daily activities on one scale. Is the patient reporting on impact? Is the patient reporting on severity? Or are we just asking for both in one single go? A good diary design will separate these constructs so that you have a clear measure, separate and clear measure. Secondly, in terms of your response options, there's an inconsistency. So if you look at the options for the headache question, it says 0 is no headache. And in the second question, in the muscle ache question, you have 0 means none present. So this kind of inconsistency confuses the participants and could lead to increased errors. And all in all, for this, the type of diary design can actually risk that you're collecting very poor quality data. So if participants aren't sure how to answer, or the different, your participants are interpreting the different questions differently, the data set will become noisy and less reliable. So we want to ensure that we keep the diary simple, we keep it consistent, and your questions are focusing on one construct at a time. And you can ask follow-up questions if you want to, but one construct at a time. And then while we're on the topic of data quality and integrity, there's something that I want to discuss here, which is using electronic diaries versus paper diaries. Next slide, please. I want to have this discussion using the study here, which is a classic study to look at why ECOA matters. So in this studies, we have two different groups. One group was asked to complete paper diaries, and the other group was asked to complete electronic diaries. For the paper diaries, participants were just asked to complete daily symptom diaries on paper. And when they were interviewed at the end, about 90% of the participants reported that they had been fully compliant and they fully completed the diaries every day on time as expected. And the interesting part for this study is that the paper diary binders that were handed over to the participants that contained the diary cards, those were fitted with light sensors that unobtrusively recorded the date and the times the binders were actually opened. So not only were we getting subjective data, the sensors revealed the objective data, which was different, as you can see, from what the participants were reporting. So the actual, in reality, compliance was only around 11%. So many participants opened their diaries once a week and then retrospectively or backfilled their entire week's entries, which we also call as parking lot syndrome. So your participant coming for a visit and sitting in the parking lot and completing all their diaries. So although they thought they were being compliant, the data that we got was actually inaccurate and far less reliable. And by contrast, you can see in the electronic diary, participants were given a palm device with prompts and built-in timestamps. So compliance was about 94%, which matches the actual compliance as well. So with paper, you're often capturing what patients think they did, as is the case in the study. But with electronic diaries, because they're date and timestamp, you get the actual real data in real time. And for your safety monitoring and your data quality for vaccine trials, this is absolutely critical. Next slide, please. And now looking at the different modality options, we can go on to the next slide. So when you're implementing clinical outcome assessments in your vaccine trials, because we deal with a large number of participants in these trials, you cannot really do a one-size-fits-all approach. We have different study populations, they're different geographical locations, infrastructures, and they all need different solutions. So often a mix of modalities is the best way to ensure that you're being inclusive in your approach and you're also getting the right data for your trial. The different modality approaches, so let's just briefly look at these, starting with provision devices. So these are devices that you would hand over to the participants, and they already have the study app loaded on them. These are highly secure, they ensure data integrity, and they also have offline capability. So this is absolutely critical when you're looking at areas with low connectivity or no connectivity at all. And then also it automates the uploads to the study database automatically. However, the trade-off is that because shipping could take time, setting up could take time, you want to build in those timelines when you're looking at provision devices. Then we have bring your own device. This is the most flexible approach. This basically means participants use their own smartphones. So it automatically reduces your costs, logistical issues. However, participants may not want to use their device, or they may not have a suitable phone, so the app might not be supported on whatever operating software they have. So for BYOD, we always recommend to use a hybrid approach, so mixing BYOD with a pool of provision devices as your inclusive strategy. Excuse me. Then you have ECOA Web. So for ECOA Web, it uses a URL access. This is device agnostic, eliminates most device logistics because it's just on the web, they type in the URL and they get access. It can be cost efficient, but this is the one modality that is heavily dependent on reliable connectivity, which can be a limitation in certain regions. And then lastly is your ECOA Live Telemedicine. So if you're using, let's say, an interview-based approach, so you're interviewing the participants for the assessments, you can use this as a telemedicine, as a live, as a remote, as an alternative to interview assessments by doing a remote data collection. So this can help offset some of your transcription errors, for example, and then some backup. Sorry, just give me one. Okay. So with ECOA Live Medicine, ECOA Live and Telemedicine, this can help improve your collection strategy. So there are no transcription gaps when you're doing the interview assessment. And then also there's audit trail because this is recorded remotely. So before you look at modalities, there are other considerations to keep in mind, such as what does the device access look like for the population you're working with? What is the network coverage, again, for the geographical location that you want to work with? So you have a participant in an urban setting, they may have a more reliable smartphone and Wi-Fi, while someone in a rural area might need a provision device that has an offline capability as well. Remember, participants should feel confident, comfortable in using the tool that is being provided to them, because that's the only way they would stay compliant. If it's too complex for them, they might just lose interest altogether. Secondly, to think about workflow flexibility. So if you're keeping your diaries available only 9 to 5, then you will lose some of the shift workers who are not working 9 to 5. So we want to ensure that we're inclusive for them as well. And then any study-specific features. So if you have vaccine trials where you're asking the participant to record the redness and swelling on their arm, for example, you could also then look at image capture as well. But again, you want to make sure that you have these tools that are specific to your study as well and your study populations. So in my experience, the mixed modality approach is something that we see the most with sponsors. So you'll have BYOD wherever possible to reduce burden, but then also keeping some provision devices on site. So if the participant's not comfortable in using their own phone or their phone is not suitable enough, then you have some backup devices available. We want to make sure that we're being flexible in our approach and how we are implementing ECOA, because we want to build a flexible ecosystem that works for everyone. Next slide, please. And then for the BYOD considerations, a couple of things I just want to highlight. If participants are using their own device, they could inadvertently just uninstall the app, decide they don't want to participate, or just simply ignore notifications. So for that one, I would recommend that we embed participant training as part of your vaccine trial, where you walk the participants through their roles and responsibilities. So why it's important to report their symptoms every day, why it's important to be compliant, and then also walk them through the study app itself, like what's expected of them, how to complete the diary. That way you sort of remove the unknowns, make them comfortable, and then they can then be engaged more. Also, secondary strategies such as email reminders or SMS if they're just simply ignoring notifications. And then also monitoring the data that you're getting every day. Next, we want to look at regulatory acceptance. So for this one, ensure that you're not only looking at younger population, we want to ensure that our elderly populations equally included, involved, because the data that you will get at the end, if you're using younger participants only, may not be a true reflection of the population where you want to implement your vaccine trials. And the last thing I want to highlight here, which is super important for BYOD, is that BYOD studies often will need extra lead time, especially when you're working with app stores. So this has to be built into your overall study timelines in order to avoid any delays. So BYOD can offer great flexibility. It can help lower your logistical costs, but there's a lot of planning and backup devices that go into ensuring that this is the best strategy for your trial. And then you also need a lot of participant support and participant engagement when you're implementing BYOD. Next slide, please. And we talked about this briefly when I was talking about the the types of symptoms you collect. And injection site monitoring is one of the key symptoms that you monitor as part of your vaccine trials, where you ask the participant if they have any pain at the injection site, if they have any redness or swelling. And for injection site monitoring here, you can also add imaging to your ECOA data collection. That just gives you a more objective time-stamped evidence. So instead of just asking participants to take a ruler, mark whatever redness it is, 2 millimeters, 3 millimeters, and then add that to your diary, this just gives an objective component, which is a timestamp collected in real time. You can again embed alarms, notifications that remind the participants at the right times to upload the picture. And then with the right system, your system can ensure that there's an audit trail as well, so you know exactly when this was uploaded. Next slide, please. And then this slide, this discussion basically brings together the regulatory perspective and what we also see in the literature. So FDA's CBER guidance basically set the foundation for how we do vaccine trials. It specifies the common local and systemic reactions that have to be monitored. These are non-negotiable, so have to be monitored in vaccine trials, such as injection site pain, swelling, fever, fatigue, headache. And the great thing about this guidance is that it provides you with a consistent framework that can help safety monitoring across your studies, across your different phases. However, again, same lesson here, that monitoring is not one size fits all. So there are extra parameters that you can add. So depending on what you've seen in your preclinical studies, for example, any biological plausibility of certain events, you want to take that into account, or experience with similar licensed products. So this flexibility ensures that the trial that you're conducting is capturing the exact relevant data for your specific vaccine. So the key point here is that your reactogenicity data from your e-diaries is monitored continuously. So you'd see often in vaccine trials, there are actually study stopping rules that are included in your protocol. So by tracking your events in real time using electronic diaries, this will feed directly into your stopping rules. So for example, if you have a cluster of grade 3 events reported by a participant, or for out of the 7 days, for like the last 4 days, participant has reported a high-grade fever. So you may need to pause the trial for a safety review. So without that real-time data capture, that responsiveness itself is not possible. So for us, when we work in vaccine trials, we want to ensure that regulators and literature and our practices are all aligned. So consistently capturing the core solicited events is absolutely essential, and e-diaries can make this very feasible for us. Next slide, please. And all the reactogenicity diaries are embedded into the FDA guidance and the EMA guidance, which I highlighted at the beginning of the session. Next slide, please. So let's start with some of the systemic reactions that the FDA and the EMA both put forward that you should be collecting as part of your vaccine trials. So these are your nausea, vomiting, diarrhea, headache, and fatigue, or myalgia. You will grade these on a scale of mild, which is 1, to life-threatening, or grade 4. For vaccine trials, grade 4 usually means the study will stop. What changes across all these different grades is not just severity of the symptom, so you also need to take into account the impact. So from having no interference with daily activities all the way to the participant require ER visit or hospitalization, you want to build that in your safety signal monitoring. Next slide, please. Same principle applies here when you're looking at local reactions, so symptoms like pain, tenderness, redness, swelling, or fever. They are measured size-wise and intensity. So for pain and swelling, you will look at the size as well. And then again, the functional impact as well. So swelling, for example, you're not only measuring this by the diameter, but then also how much it is affecting daily activity. And what's important, or the key takeaway for the guidance slide here, is that the e-diaries are designed to align directly with these grading scales. So your response parameters should include this grading scale as well, to collect data that is consistent, interpretable, and submission ready at the end. And because your e-diaries are collecting symptoms daily, your investigators, your site staff can monitor these reactogenicity data that you're getting in real time and continuously. So, and if any event reaches certain thresholds, such as a cluster of grade 3 fevers that I gave an example of, this can contribute to your protocol-defined stopping rules. And again, this gives your sponsors, the regulators, confidence that the trial is being looked at in a safe way. So this isn't just, again, nice to have or good to look at. These are the foundation of how you design your e-diary questions, how you look at symptoms and how you grade them, and how we protect our participant in vaccine trials. Next slide, please. And then just looking at the evolving regulatory landscape, there has been some guidance on how you design vaccine trials, how to collect your data, how to submit your data as well, and what analyses to carry out. These specific ones I want to highlight are the 2019, this was a critical piece that was introduced, so guidance specifically on submitting data sets for vaccines to the Office of Vaccines Research and Review. This is aimed at reducing submission bottlenecks, emphasizes on standardized electronic formats, and in turn, again, support better use of participant-reported outcomes across studies. There's been a strong emphasis recently on incorporating digital health technologies and patient-focused drug development guidance. So the FDA's 2021 draft guidance explicitly calls out the need for digital tools that are not just digital, but they're fit for purpose as well. They're accessible and they're inclusive. You want to ensure that you're catering to a diverse population in your trial. And then for accessibility, you can either do translated versions of the questionnaire, use larger fonts, or simplify your user interfaces as well. And then for the patient-focused drug development guidance and the updated EMA guidance for clinical evaluation of vaccines, this underlines the expectations clearly. So for your clinical outcome assessments, your patient-reported outcomes, how these should be integrated into your trial endpoints and are part of your regulatory decision-making. Next slide, please. So when we're talking about regulatory considerations in vaccine trials, there are two sides to the challenge. There's the regulatory environment itself, and then there is the ethical standards. Focusing on the ethical standards, this must be at the heart of your trial design and conduct. So again, ensure that you've taken informed consent from your participants, protect their privacy, and then again, monitor safety rigorously. These are all essential. These safeguards can also add complexity and extend your timelines. However, it's important that you anticipate these early at the beginning of the trial. But with both your regulatory environment and your ethical standards, if you do careful implementation planning, built-in time for your regulatory reviews, design tools that meet the international ethical standards, and proactively plan for country-specific challenges, this can help significantly reduce your risk. So what we want is we want a thorough, proactive implementation plan which will allow your trials to stay on track while still upholding the highest regulatory and ethical standards. And I want to conclude my presentation on the last slide by sharing a case study which I'm presenting as part of the World Vaccine Congress Europe. And for this one, I looked at barriers and enablers. So Sub-Saharan Africa is a region which is not really tapped into. So we want to look at what are the different barriers that we can address in order to ensure that we are not leaving this strata of population. And then also enablers. So what are the things that are already present that we can tap into and then further focus or zoom in on to do vaccine trials in sub-Saharan Africa? So just some of the barriers I want to highlight. There's of course the urban-rural divide, where you have adults in rural areas that are nearly 50% less likely to have internet as compared to your urban counterparts. Mobile broadband consumes about 4% of household income compared to a 2% global benchmark. Literacy and languages are also an issue, and gender inequality, where there's at least a 10% gap in internet usage between men and women. So women tend to use less internet. However, not everything is bleak. There's certain enablers that we can use, so we can sort of build further on it as a strong foundation. So there's network coverage. Over 90% of the countries that I looked at for this research have at least 75% 3G coverage already. So that means the infrastructure already exists. Provision devices, so we don't have to ask participants to use their own device. We can give the participants devices with offline capabilities. Audiovisual consent tools can be used for interview-assisted approaches. And then again, literacy, because it's a driver in my research, it showed as the highest or the strongest correlation with all the different enablers. So addressing literacy can help strengthen the feasibility of your clinical trial. So the key takeaway that I want to conclude the case study with and the slide with is that all these enablers that exist, they don't exist in isolation. So infrastructure, affordability, literacy, and gender equity are all interconnected. So what we want to do is we want to implement a holistic strategy for our COAs. We cannot solve connectivity without solving affordability. And then again, your participation will be skewed otherwise. If we only provision devices but then ignore the language needs, your participants will still struggle and they will not engage with you. So designing trials that address these factors and ensure that they are scientifically sound, but also ethical and inclusive and representative can help build you a very successful vaccine trial. Thank you. I will now hand over to my colleague, Vivian Chen.
Hi, thank you, Simon. Next slide, please. As Dr. Rudo mentioned earlier, vaccine development has unique challenges. Vaccine trials often involve the general healthy population, plus there's a low tolerance for safety concerns, especially cardiac safety concerns. Although vaccines have been used primarily to prevent infectious diseases, they are also starting to be developed for non-infectious diseases. And this can include some types of cancer, elevated lipids, and Alzheimer's disease, for example. These studies involve patient populations that are higher risk for cardiac safety events, and this can obviously affect the level of cardiac monitoring that's required for these programs. There are a broad range of endpoint solutions available for cardiac safety monitoring, and these include monitoring cardiac adverse events, cardiac labs such as troponin and inflammatory biomarkers, electrocardiograms or ECGs, and cardiac imaging, which includes typically cardiac echo and cardiac MRI. Next slide, please. Next slide. Cardiac adverse events due to vaccines are rare, but they do occur and they can be serious. Just starting off with commonly recommended vaccines such as Tdap, tetanus, diphtheria, and pertussis, and the varicella zoster vaccine, there are no known increased cardiac risk of cardiac events for these vaccines. However, for the smallpox vaccine, which is no longer recommended, there are cases of rare myocarditis. For vaccines that utilize an adenoviral vector, there can be rare thrombotic thrombocytopenia. And as I mentioned, as we heard, messenger RNA COVID-19 vaccines can cause a rare myocarditis and also pericarditis. This typically is more common in adolescent males. Typically occurs often within 7 days after the second dose. There are also cases of rare Takotsubo cardiomyopathy, which is a heart failure. This occurs more frequently in females. And for the most part, for vaccine-induced types of Takotsubo, these patients recover. Next slide. For myocarditis, the clinical trial assessment is as follows. The symptoms that can include acute chest pain, shortness of breath, and palpitations. There can be ECG findings or abnormalities, such as tachycardia and ST wave changes. There can be frequent atrial ventricular ectopy. For blood tests, we can see elevated cardiac troponins and elevated inflammatory biomarkers. And for cardiac imaging, using echo or MRI, there can be abnormal cardiac function, wall motion abnormalities, and also for MRI, late gadolinium enhancement. This is a contrast agent indicative of scarring in the heart, and there can also be myocardial edema. Next slide, please. Safety ECG monitoring is critical in clinical trials. Shown on the bottom left is a traditional 12-lead ECG device. On the bottom right is a home ECG with an accompanying smartphone. And we also recommend that central ECG lab also be used for ECG monitoring in clinical trials. A central ECG lab collects and analyzes and stores ECGs. This reduces the measurement and interpretation variability of these ECGs, and this can result in a lower false positive rate for safety signals. Next slide is an example of an ECG. Next slide, from a patient with acute myocarditis following a COVID-19 vaccination. The asterisks on the ECG note ST elevations in the lateral leads, 1 and AVL, and reciprocal ST depression in the V1. Next slide also is an example of cardiac imaging techniques. Echo and cardiac MRI are commonly available imaging modalities. They're both non-invasive. They don't involve radiation. The echo is more widely available and lower cost. The MRI, however, provides a higher resolution. It is more expensive and can be more burdensome for trial subjects, and especially for those with claustrophobia. The next slide, please, illustrates an MRI from 2 patients who developed acute myocarditis after a COVID-19 vaccination, patient 1 and patient 2. The yellow arrowheads point to areas of LGE, or late gadolinium enhancement, corresponding to scar tissue in the myocardial walls. And for COVID-19 vaccination, for those patients who develop myocarditis, this can persist chronically as well. Next slide, please. Next slide. Oh. In summary, the cardiovascular safety profile of all vaccines should be thoroughly characterized. There's a low threshold for cardiovascular side effects for vaccines, especially if the vaccine is intended for the general healthy population. The level of cardiac safety assessment should be individualized for the vaccine program, and important determining factors include the patient population, any mechanistic and preclinical cardiac safety data, and any available clinical data in the program. It may be the case that the incidence of cardiac safety events is very low, and in this case, a post-approval cardiac safety evaluation may be required by health authorities. For all vaccine programs and all clinical programs, there is a regulatory requirement for transparent safety reporting and long-term follow-up for any cardiac safety events or issues. So I think this is my last slide.
Great, thanks. So thanks to Dr. Chen, thanks Saima, definitely for your really quite valuable insights there. That does take us to the conclusion. I'm going to purposely skip this summary, recap slide here and ask the two presenters, SMEs, to come back on screen here. We're going to try and squeeze in. You've been great about sending in questions, and I want to try and get to them, or at least a couple of them here if we can. So I'm going to jump right to that. But again, appreciate everyone's time and attention here, certainly. If anything we don't get to here, by the way, we will certainly reach out to you directly and follow up. Simon, if I could direct this first one to you, it's about compliance rates. And the question is, what compliance rate do you typically see within vaccine e-diaries? And what can you do to ensure high compliance in a study?
That's a good question. So what I've seen so far, and in my experience, compliance with e-diaries has usually been quite high. And I actually looked at some vaccine studies that we've done here at Clario, and I reported a compliance of about 89 to 97% across phase 1, 2, and 3 vaccine studies. So looking at the different elements of how we did those vaccine studies, I want to say I attribute that compliance or high compliance to a combination of strategies. So we looked at the right timing of your assessment, so making your availability windows much longer. So you want to ensure that you're capturing more participants that way. And then setting up reminders on the app. So these are different from your phone reminders. Just on app, you have visible icons that appear. So reminding the participant that, okay, the diary is available now for them to complete. And then also training the participants. So you want to ensure that the participants fully understand what's expected of them, or but then also walk them through how to report the symptoms, what are the different types of questions that they will be answering. That just helps ensure that they are engaged throughout your study trial. And then lastly, you can also use gamification, which we have here at Clario. So this is just to keep the motivation high. So it's basically just giving them a star every time they complete a diary, and at the end of the 7 days, they see 7 stars. So just sort of like elements like that can help really drive up compliance.
Yeah, I think that's great. I mean, certainly that's an innovative approach, and as you said, sometimes the simplest bit, the participant training and the importance of that. I think if we don't forget that piece, we can actually make a pretty big impact on compliance there as well. Dr. Chen, one for you, if I could, kind of specific question, but should all vaccine trials include a baseline and an on-treatment safety ECG?
There's no regulatory requirement for ECG performance in vaccine trials, but it would be very useful and reasonable to perform a baseline. The baseline is useful for comparison if there are any future adverse events during the trial and after the trial. It is important to consider an on-treatment ECG based on any earlier phase or non-clinical safety signals. So the answer is it is very useful to have an ECG in the clinical trial.
Yeah, especially at baseline. Okay, great, thanks. Well, unfortunately, although there are more here, I'm going to have to cut us off in the interest of time. Thank you again to our audience for joining and such engagement here. Thanks to Saima and Fabian, certainly, for your important contributions here. Sonia, I will turn back over to you now if you can close us out.
Oh, certainly will do. Thank you very much, Todd, Saima, and Fabian for answering all those questions. Just like Todd said, we are closing out now the Q&A portion of the webinar. So thank you very much for sending in all those questions. Now, if you couldn't attend to your questions, remember the team at Clario will follow up with you after this presentation. And if you are interested in hearing more from Clario, and who wouldn't be, please check out their next webinar, which is scheduled for Wednesday, November the 12th. Every Patient Matters, Bespoke Digital Endpoint Strategies for Rare Disease Drug Development. And I put the link in the chat box already, along with the link with today's recording that you can share with your colleagues. Now, as I said before, if we could not attend to your questions, rest assured the team at Clario will follow up with you. And if you have any further questions, please direct them to the email address you see there on your screen. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of today's event. And as I mentioned, that link is in the chat box there for you to review for today's webinar. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event to gain access to features such as viewing the recording and even more. Let me see here now. Now, I just wanted to say that there are no handouts. So just wanted to say thank you very much, everyone, for participating today. And I just want to say thank you very much to Todd for being my co-moderator, for Saima and Fabian for great presentation. We hope you found this webinar informative. It has been my pleasure to be your moderator for today's event. On behalf of the team here at X-Talks, we thank you for spending your time with us here today. Until the next time we meet, please take care and bye for now. Bye, everyone. Bye, Todd. Bye, Sama. Bye,
Thank
Fabian.
you.
Thank you all. Thanks.
Bye.